Okay, let's start with a little word association. If I say GLP-1 or, maybe Ozempic or Wegovy, what's the absolute first thing that pops into your head?
Weight loss. Almost certainly weight loss. Maybe diabetes if you're in the know.
Exactly. It's the cultural phenomenon of the decade, right?
Hmm.
We think of these as the drugs that shrink waistlines, quiet the food
These
noise.
celebrities look suspiciously gaunt on the red carpet.
Right. It's effectively the diet pill that, well, that actually worked.
That's fair. It's completely dominated the conversation around metabolism and obesity for the last few years.
But, and this is where it gets really, really interesting and why we're doing this deep dive, what if I told you that weight loss might actually be the least impressive thing these drugs are doing?
That is a bold claim. I mean, considering how effective they are for weight loss, but the data is definitely starting to back that up.
I know. So we're looking at a brand new paper just published last month, December 2025, in Cell Metabolism. It's from a team led by Huang and colleagues, and it just completely flips the script. They aren't looking at these drugs as diet pills. They're looking at them as comprehensive, body-wide, anti-aging technology.
And we're not just talking about, feeling younger. We're talking about reversing the molecular clock across the entire body.
The whole body, from the heart to the brain to the muscles. It's mind-blowing stuff.
It is.
So our mission for this deep dive is to really unpack this study. It's titled Body-Wide Multi-Omic Counteraction of Aging with GLP 1R Agonism. We're going to look at three specific layers.
Okay.
First, the physical proof, how these drugs stop frailty. And spoiler alert,
Yeah.
it has nothing to do with losing weight.
Right. Then we'll get into the molecular evidence,
Yeah.
the multi-omic deep dive that shows biological age literally reversing in the organs.
And finally, the mechanism. And honestly, this is the part that made my jaw drop.
Right.
It turns out the brain might be the remote control for the entire aging process of the body.
It's the brain-body axis of aging. It's a fascinating concept that, it really challenges how we think about getting old.
So let's dive in. The setup here is crucial. The researchers didn't use humans. They used male mice. But not just any mice.
Right. They started with middle-aged mice, about 11 months old. If you want to map that to human years, you should think of someone in their 40s, maybe early 50s.
Okay.
And they treated them with a drug called exenatide. Now, just for clarity, exenatide is a GLP-1 receptor agonist. It's an older one compared to, say, semaglutide, but the mechanism is the same. It mimics that GLP-1 hormone.
So they treated these middle-aged mice for 30 weeks, which brings them well into what you call old age. But here is the critical detail that sets this study apart. They used a low dose.
This is key. Usually in these studies, the treated animals lose a ton of weight because the drug makes them stop eating.
Right, caloric deficit.
They go into a caloric deficit. And we know for a fact that caloric restriction extends lifespan. So if the mice live longer or look younger, the critics would just say,
Well, they ate less. It's just a diet.
exactly. But in this study, the dose was low enough that the treated mice did not lose significant body weight, not compared to the control group. They ate about the same amount.
So what we have here is a clean experiment. They've isolated the variable.
Precisely. If the mice got healthier just because they were skinny, you could argue it's just from carrying less fat. But here, the weight was the same. So any anti-aging benefit we see, it's coming from the drug's direct effect on the biology.
Not from weight loss.
Not from weight loss.
Okay, so we have these old mice on a low dose of GLP-1. They aren't skinny supermodels. They look like normal old mice. But physically.
Oh, they were veritable athletes compared to the control group. The difference was stark.
What did they test?
So the first thing they looked at was grip strength. As mice and humans, for that matter age, they get frail, they lose muscle power.
And that's a huge predictor of mortality in people, right? Like, can you open a jar?
A huge one. If you can't open a jar or grip a railing, your risk of death goes way up. The control mice, they followed that expected decline. They just got weaker over time.
But the treated mice.
Maintained their grip strength. They didn't get frail. The line it flatlined that decline.
That's huge. Frailty is basically the beginning of the end for so many elderly people. If you could just stop that.
Exactly. And it wasn't just raw strength. They did a test called the rotarod. Picture a treadmill, but it's a rotating log that the mouse has to balance on.
It's like a lumberjack competition for mice.
Ideally, yes, it spins faster and faster. It tests coordination, balance, motor learning.
And the old, untreated mice were bad at it.
Terrible. They fell off pretty quickly. The GLP-1 treated mice, though, performed significantly better. Their motor coordination was preserved.
And I saw they also tested anxiety.
They did. It's called an open field test. Mice are prey animals, so naturally they get anxious in open spaces. They tend to hug the walls.
Phygmotaxis.
Phygmotaxis, yes. The treated mice spent significantly more time in the center of the arena. They were less anxious, more willing to explore.
So we have stronger, more coordinated, and more chill older mice. But I want to be fair to the source material here. This wasn't a limitless pill, right? It didn't fix absolutely everything.
No, and that's an important nuance. They ran cognitive tests mazes to test spatial memory, like the Y maze. And in this specific cohort, the drug didn't produce massive improvements in, say, learning speed.
So it's not necessarily making them geniuses.
Right. It seems to be preventing decline in specific aging vulnerable pathways like frailty rather than being a universal performance enhancer.
Speaking of performance enhancers, there was another control group that I found fascinating. They gave the same drug to young mice.
This is one of my favorite parts of the study. They took 3-month-old mice, basically young adults, and gave them the drug. And guess what happened?
Nothing.
Almost nothing. No grip strength improvement, no cognitive boost. It essentially had no effect.
That is so telling. It means this is a restorative therapy. It's
Yeah.
like if your car is brand new, changing the oil doesn't make it faster.
It just runs like it should.
But if your car has 200,000 miles on it, that maintenance is the difference between running and breaking down.
That's a great analogy. It counteracts the damage of aging. If there's no damage yet, there's nothing for it to fix.
Okay, so physically, the mice are younger. But the researchers didn't stop there. They decided to pop the hood and look at the engine.
By engine, you mean the cells.
I mean the cells. This is where we get into the multi-omic part of the title.
Right. Multi-omics sounds like jargon, but it just means they analyzed multiple layers of biological data at the same time. Specifically, the transcriptome and the DNA methylome.
Let's unpack those terms so we don't lose anyone, because this is where the real proof is.
Okay, so think of your DNA as the hard drive. It has all the files, all the instructions to build a bottle.
Got it.
The transcriptome is like the list of programs that are currently open and running, which genes are turned on or off. That's the RNA. Then you have DNA methylation. That's like the permission settings on those files, little chemical tags that tell the cell, do not open this folder or read this file constantly.
And as we age, those permissions get messy. The wrong programs start running. The hard drive gets fragmented.
Exactly. So the researchers took tissue samples from all over the body, the hypothalamus in the brain, the frontal cortex, fat tissue, the colon, the heart, skeletal muscle.
So this wasn't just checking the pancreas or the gut.
No, this was a systemic audit. And they used this technique called principal component analysis to visualize all that data. Imagine a chart where every dot is a mouse.
Right.
All the young mice are clustered in one corner, let's say bottom left. All the old control mice are way over in the opposite corner, top right. Biologically, they are miles apart.
I'm visualizing it.
Now, when they plotted the old mice that were treated with the GLP-1 drug, their dots physically shifted on the chart. They moved away from the old cluster and migrated back toward the young cluster.
They literally reversed the data signature of aging.
In almost every single organ they tested, the hypothalamus, the heart, the muscle, the drug, reversed the gene expression changes that are associated with aging.
And what exactly were these genes doing? What specific programs were being reset?
They found reversals in 3 major aging bad guys. First, cellular senescence.
The zombie cells.
The zombie cells. They stop dividing, but refuse to die, and they just pump out toxic signals. The drug turned those signals down. Second, inflammation, what we call inflammaging. Reduced. And third, autophagy.
Autophagy is the cell's cleaning crew, right?
Right? It literally translates to self-eating. It's how cells recycle their own junk. Aging shuts this down and trash builds up inside the cell. The GLP-1 agonist turned the cleaning crew back on.
So, less inflammation, fewer zombie cells, better taking out the trash.
Pretty much. And it wasn't just the RNA. The DNA methylation, those permission settings we talked about, also showed a reversal of epigenetic aging, especially in the brain and the skeletal muscles.
And that's huge because those epigenetic clocks are our best measure of biological age.
Right. They're the most scientifically accurate way we have.
Okay, so the body's getting younger. But here is the trillion-dollar question. How? How does a drug that mimics a gut hormone tell the skeletal muscle and the heart to stop aging? Is it? Is it going to each organ individually?
That is exactly what the researchers wanted to know. Is this a local effect or is there a central command? And this leads us to the real aha moment of the paper.
The brain-body axis. This experiment was it was wild. Walk us through what they did.
So they hypothesized that the brain, specifically the hypothalamus, might be coordinating this whole thing. The hypothalamus is known as a master regulator. It controls metabolism, temperature, hunger.
So they decided to sabotage the hypothalamus.
In a very clever way, they used a virus to deliver a genetic tool that silenced the GLP-1 receptors only in the hypothalamus.
Okay, let me get this straight. The mice still got the drugs systemically. It was circulating in their blood.
Yep.
So it could still hit the heart, the pancreas, the muscles directly. But the brain couldn't see it.
Precisely. They essentially blindfolded the hypothalamus to the drug, and then they looked at the anti-aging effects in the rest of the body.
And what happened?
The effects collapsed. When the brain couldn't see the drug, the anti-aging benefits in the heart, in the skeletal muscle, in the blood cells, they were severely weakened or just gone completely.
That is unbelievable. So even though the drug was literally touching the heart cells, the heart didn't get younger unless the brain gave the order.
That appears to be the case. It suggests that the GLP-1 agonist stimulates the hypothalamus, and then the hypothalamus sends out a signal, likely through the nervous system or hormones, that tells the rest of the body, repair yourself, reduce inflammation, turn on autophagy.
It's like the hypothalamus is the general and the organs are the soldiers. If the general is asleep, the soldiers don't know the war is over. They just keep acting old.
It fundamentally changes how we view aging. We often think of aging as this, wear and tear in each individual part. Your knee wears out because you used it too much. But this suggests there is a driver. And if you wake up the driver, they can steer the car back onto the road. The brain is the master switch.
That puts a whole new spin on mind over matter. But before we get too philosophical, we have to talk about the competition. In the world of longevity science, there's one reigning champion. The heavyweight title holder. Rapamycin. It's an mTOR inhibitor. For listeners who don't follow this stuff, mTOR is a protein that tells cells to grow. Rapamycin shuts it down, putting the cell into a repair state.
It's the gold standard in the lab. If you want a mouse to live longer, you give it rapamycin.
So how does our challenger, the GLP-1 agonist, stack up?
Well, the researchers ran a head-to-head matchup. They took older mice, starting at 18 months this time, so truly elderly, and gave one group exenatide and the other group rapamycin.
And the winner is?
It was a draw, a shockingly close draw. When they looked at the molecular data, the gene expression, the DNA methylation, the two drugs produce highly correlated changes.
So they're doing almost the same thing.
They seem to be converging on very similar pathways. There were some nuances, though. Exenatide appeared slightly better for skeletal muscle health. Rapamycin was a little stronger in the frontal cortex and in the circulating metabolome.
That's fascinating. So the weight loss drug is essentially punching at the same weight class as the most potent anti-aging drug we know of?
Yes. And you have to consider accessibility. Rapamycin is an immunosuppressant. It has significant side effects. GLP-1 agonists are already approved for millions of people. This puts GLP-1s firmly at the table as a Tier 1 longevity intervention.
It really reframes everything. We see all these headlines about Ozempic face or people worrying about muscle loss during rapid weight loss.
Which is a real concern if you don't eat enough protein.
Of course. But this study, it suggests that underneath all that, at a molecular level, the drug is actually trying to save your muscle.
Exactly. The frailty data we talked about earlier supports that. In the absence of starvation, the drug is preserving muscle function. It suggests that if patients eat properly and maintain activity, these drugs could be incredibly protective.
So let's bring this all back to the listener. What does this all mean? We've established that GLP-1s preserve strength, they reduce frailty, and it's independent of weight loss. We know they reverse molecular age across the whole body.
And we know the brain is the control center for this whole process.
The implication is that we might be treating aging as a coordinated biological program that can be hacked. We aren't just patching holes in a sinking ship.
We are finding the command that reverses the engines.
And it makes you wonder about the future of therapy. This study used a drug to stimulate the hypothalamus. But if the hypothalamus is the key...
That's the big question. If the hypothalamus is the remote control that tells the body how fast to age, do we even need systemic drugs?
Right. Could we use non-invasive brain stimulation? Could we just tickle that specific part of the brain with ultrasound or magnetic fields and trick the body into staying young without ever taking a pill?
It's science fiction territory, but this paper builds the launch pad for it. If we can map the exact signal the hypothalamus sends to the heart, maybe we can bypass the drug entirely.
Or maybe we find out that certain behaviors, meditations, specific types of exercise, deep sleep, are actually natural ways of pressing that same button in the hypothalamus.
It's entirely possible. The hypothalamus is responsive to our environment. It integrates stress, nutrition, light. This study just reinforces that keeping your brain healthy is arguably the most important thing you can do for your body.
So next time you see a headline about a celebrity shrinking away on a GLP-1 drug, try to see past the vanity. You might be looking at a crude, early version of a technology that's accessing the body's master control switch for aging.
It's a weight loss drug by accident. It seems to be a neuroprotective, anti-aging agent by design.
And on that note, we'll leave you to ponder. If your brain decides how old your body is, how do you convince your brain that you're actually 25?
Still working on that one myself.
Thanks for listening to The Deep Dive. We'll catch you on the next one.